interested in transcription?
That was when I was an undergraduate student, basically, and got a lot of lectures on molecular biology and really enjoyed understanding more about it, and this sort of what you could call machines on genes type things, and got me fascinated, and that's how I got interested in, studying gene regulation and the machinery that's involved in transcribing genes. And so that's how I got into transcription, which is that process that makes RNA from DNA.
Yes, thank you.
And what did you expect to find? Were there any surprises?
So if it relates specifically to the study, yes, then yes, there were a few surprises, because initially we, as I mentioned earlier, we had one PhD student, my first PhD student, who basically made a few mutations in residues that we thought are very important in coordinating zinc ions in this protein. And it totally killed the whole activity of this protein complex, not just that protein, but the whole protein complex didn't function. So that was a surprising result. And then we dug deeper and deeper and found that the protein was still nicely incorporated into the complex. The complex still bound DNA. That was still a surprise as well, because we thought initially zinc fingers. We thought if it's a zinc coordinating amino acids, then if we mutate those, we thought we might mutate a zinc finger and therefore not have DNA binding. But it wasn't a zinc finger. It turned out to be a slightly different variant that also is involved in coordinating zinc ions, but it had nothing to do with DNA binding. So that was a surprise. And then we dug deeper and then found that even the recruitment of the polymerase wasn't affected. So all in all, the mutations gave a very dramatic phenotype, and yet we couldn't pin it down to any of the obvious defects in assembly of the complex, recruitment of the polymerase, DNA binding. They were all fine. And it turned out that actually there was a defect after all these events happened. So it binds DNA fine as a properly assembled complex. It recruits the polymerase, but then there is a problem. And that was actually also what led to what is a major hurdle in our analysis. It was to actually then prove we knew where the defect wasn't, but we didn't know where the defect was exactly, and to prove that was actually very difficult.
Okay. How did the results from this paper enhance the field?
Well, because so we've been long wondering about. We knew that there was a TF2B-like molecule involved in Pol3 transcription. There was a TF2B molecule involved in Pol2 transcription. And so it made sense, because given the conservation of all these mechanisms and processes, that there might be a similar protein involved in Pol1 transcription. And so identifying that this protein, TAF1B, was the structural and functional homologue of TF2B and TF2B-like molecules in Pol3 transcription, provided a sort of a missing link in understanding, how these machineries have such conserved mechanisms have indeed a complement of this protein also in Pol1, and that was the sort of missing link there. So it was, from that sense, it was quite an interesting finding. Yeah.
Well, I think you've already answered the next question we had, which was, were there any challenges?
There are always challenges in research, yes. So there's nothing new there. So there are stages in which things will go all very well, and then you could hit a hurdle, and it could be either a hurdle because it's technically very challenging, or you haven't quite answered the question the right way to get further. Or it could be that it's not a technical challenge, but your whole, indeed, when you ask that question, your whole design of the experiment is based on a hypothesis that you have. And so, we thought, okay, well, it must be a defect in DNA binding, right? So you go and look for that, and then you don't find that. So that's so you think, well, what is it then? So you start by eliminating all sort of possibilities, and then can narrow it down. And so it's a combination of asking the right questions, and there are always technical challenges as well, because once we arrived at where we knew it was where the defect was, it was actually technically almost impossible to prove that there was a defect in that particular step. But by eliminating all the other possibilities, we had narrowed it down, and we knew it had to act there, but we couldn't provide the actual evidence for that. So yeah.
And our last and final question is, have you taken this research any further?
Yes, we have been looking at this particular protein within the complex, but also this protein in the context of other processes. So, and the way to do that is to do, for instance, more genome-wide studies where we've looked at whether this protein could be binding, because we know as part of SL1 it binds to the ribosomal DNA sequences, but actually by deep sequencing we found that this protein also binds elsewhere in the genome and may also regulate genes out with the normal rDNA repeats. So that's a completely new area that we have to explore further and find out what it's doing there. And the other way is to also find out how is this protein and the protein and part of this complex, how it is regulated. Is it at the receiving end of lots of signaling cascades, and therefore proteins that may interact with it, modify it, etc. And also to see whether it's perhaps part of other cellular processes. And so is it integrated, it's in the nucleus acting on transcription, but it is of course in the context of all these other processes that happen in the nucleus. And in order to do that, we've embarked on sort of more proteomic analysis where we're trying to identify what proteins are associated with the complex. So we've been doing lots of mass spectrometry of factors that are associated with the complex of interest, and have identified a whole new set of proteins that might interface with our complex called SL1. So, yeah.
Thank you very much.
All right. Well, you're welcome.
Thank you.
Thank you.
Is that